Welcome to the deep dive. Today we're plunging into a really thought-provoking area of medical science, gain-of-function, or GOF research. Now, this isn't about, creating comic book villains or anything like that.
Right, not at all.
It's more about scientists making specific genetic changes to organisms, tweaking them to see what happens to their biological abilities, like how easily a virus spreads or maybe how harmful it could get.
And the core idea behind it, it's really about trying to anticipate future infectious disease threats, get ahead of the curve.
Exactly. By understanding how these pathogens could evolve, the hope is we can then develop the medical tools we need, things like vaccines and treatments, maybe before a pandemic even starts.
That's right. And, well, while that potential to protect public health is obviously a huge motivator of this kind of research, it naturally sparks quite a bit of debate.
Yeah, I can see why.
Because when you're essentially, dialing up the capabilities of a pathogen, it brings up really important questions about biosecurity.
Like keeping these things locked down tight.
Exactly. And biosafety, making sure nothing accidentally gets out of the lab.
Right.
And because of these sort of dual potentials, good and bad, some GOF research gets classified as dual-use research of concern, or DURC.
GURC.
And that label means it gets extra levels of scrutiny.
Okay, let's unpack this DURC label just a bit. So it means the research could be used for good things, like figuring out how a virus might jump from animals to humans.
Beneficial purposes?
But also potentially for harmful stuff. Is that the idea?
That's the core of it, yes. And that designation triggers extra oversight. You've got institutional DORC committees, the NIH's Recombinant DNA Advisory Committee here in the U.S.,
Okay.
and similar groups elsewhere, like the EU's Dual Use Coordination Group, all get involved.
Gotcha. So our mission today is really to dig deep into the complexities of GOF research based on the material we've looked at.
Right.
We're going to look at some key examples. What does this work actually look like? We'll try to weigh the potential upsides against the risks, which seem pretty significant.
They can be, yeah.
And explore the ongoing discussions around it all, because this is a field with, massive implications, wouldn't you say?
Absolutely. Huge implications.
Yeah.
Both for our ability to maybe prevent the next pandemic,
Which we definitely want.
and the potential for things to go wrong, those unintended consequences.
Yeah.
So it's really crucial for us and for you listening to try and grasp the underlying science, the different perspectives on risk versus benefit, and the policies that are trying to govern this whole complex area.
Okay, so let's ground this conversation, maybe start with some real-world examples of GOF research, just to get a better handle on what we're actually talking about.
Oh, we do.
One set of studies that always seems to come up is that work from around 2011. Yoshihiro Kawaka at Wisconsin-Madison and Ron Fauchier over at Erasmus in the Netherlands. What were they looking into?
Right, those studies got a lot of attention. So what's interesting is that both of these teams, working independently, were focused on the H5N1 avian influenza virus, bird flu.
Bird flu, okay,
Yeah.
Which doesn't usually spread easily in people, right?
Exactly. It typically doesn't infect humans well.
Yeah.
But they wanted to understand how it could potentially gain that ability. What changes would allow it to trigger a human pandemic.
Okay, so how did they test that?
Well, they essentially played a kind of accelerated evolution game in the lab. They used ferrets.
Ferrets. Why ferrets?
Ferrets are actually a really good animal model for studying human flu. Their respiratory systems are quite similar to ours in some key ways. So they took the bird flu virus and infected ferrets with it.
Okay.
They'd let the virus multiply in one ferret, and then they'd take the virus from that ferret and transfer it to another one. And they just kept repeating this process. Serial passaging, it's called.
And what happened? Did the virus change?
It did. Over these repeated passages through the ferrets, the virus gradually adapted to its new host. It picked up specific genetic mutations.
Mutations that helped it.
Yes, mutations that allowed it to replicate more effectively in the ferret's respiratory system, which is cooler than in birds. But the really significant outcome, the headline grabber, was that the modified virus eventually gained the ability to transmit between ferrets through respiratory droplets.
So, like coughing and sneezing, airborne transmission.
Essentially, yes. Airborne transmission between the ferrets. And the key insight wasn't just that H5N1 can become transmissible in mammals,
Right.
but that it seemed to only take a relatively small number of genetic changes to make that happen. That really highlighted how unpredictable viral evolution can be and why understanding these potential pathways is, potentially important.
Okay, wow. That does sound significant. Now, jumping forward a bit to May 2013, There was another interesting set of experiments, this time led by Hualan Chen in China. What was their focus?
Chen's group was looking at something slightly different but related. They were investigating what might happen if a flu virus that already circulates in humans, they used the 2009 H1N1 pandemic strain.
Okay, swine flu.
Right. What if that virus infected the same cell as an H5N1 bird flu virus? This happens in nature sometimes.
Like a co-infection.
Exactly. And their experiments, which they stressed were done under strict biosafety conditions, showed how this process called gene reassortment could occur.
Reassortment, like shuffling the genetic deck between the two viruses.
That's a great way to put it, shuffling the genetic material. And they found that this shuffling could create new hybrid H5N1 viruses that were much more easily transmissible in mammals. They used guinea pigs in their experiments.
So this wasn't just about the bird-to-human jump scenario, but it also flagged risks in, like, agriculture, if this mixing happened on farms.
Precisely. Their work really highlighted the potential for new, possibly dangerous flu strains to emerge in places like farms where different flu viruses might co-infect animals like pigs or poultry. And it's worth noting, this research came out just before a sort of voluntary pause on certain types of H5N1 research was agreed upon by many scientists, so it added to the ongoing debate.
Right. And was this reassorted virus more deadly?
Interestingly, no. Similar to the ferret studies, the reports indicated that these modified viruses in Chen's experiments were actually less deadly in the animal models they used.
Okay. So transmissibility increased, but lethality seemed to decrease in those specific models.
In those models, yes. It
Yeah.
shows how complex these changes can be.
Definitely. Now let's move to more recent times. October 2022, a preprint study from researchers at Boston University. This caused quite a stir involving a chimeric SARS-CoV-2 virus. What exactly did they do?
Right, this one got a lot of media attention. They essentially took the spike protein, the part of the SARS-CoV-2 virus that grabs onto our cells.
But the part everyone focused on for vaccines.
Exactly that part. They took the spike protein from the Omicron variant,
Yeah.
which was known to be highly transmissible but generally less severe, and they combined it with the genetic backbone, the rest of the virus, from an earlier ancestral strain of SARS-CoV-2, which was known to be more severe, at least in certain models.
So like mixing and matching parts of different variants.
Pretty much, creating a hybrid, or chimera. Then they studied this new chimeric virus in mice, specifically a type of mouse, often used because it's susceptible to severe COVID-19.
And what did they find? This is where the headlines came from, right?
Yes.
Oh.
The key finding reported was about mortality in these specific mice. The original ancestral strain they used killed 100% of the mice in their experiment. The Omicron variant, on its own, caused no deaths in these mice.
Okay.
But the new chimeric virus, the one with the Omicron spike on the old backbone, resulted in about an 80% mortality rate in these susceptible mice.
Right, that 80% kill rate figure, that definitely caught people's attention and, fed into the ongoing debates, especially around COVID origins.
It absolutely did, but there's some really important context that often got lost in those initial headlines.
Like what? What was the missing context?
Well, first, the researchers themselves pushed back, clarifying that their specific work wasn't directly funded by the NIH, despite some early reports suggesting it was. They also disputed that it should even be classified as gain-of-function research under the existing definitions.
Okay, that's one piece.
Second, and this is crucial, while 80% mortality sounds incredibly alarming, it was actually less lethal than the original ancestral strain they used as a comparison point in the same mouse model. That strain killed 100% of the mice.
Ah, I see. So compared to the original virus in that specific experiment, it was less deadly, not more.
Correct, in that specific context. Also, many experts who looked at the study pointed out this type of research trying to understand which parts of a virus contribute to disease severity, structure function studies, is actually a fairly standard scientific practice. And it was done under established safety protocols in a high containment lab.
So not necessarily unusual research, just maybe communicated in a way that caused alarm.
Potentially, yes. And fact-checkers also stepped in to provide that context, reiterating that the 80% figure applied to specific lab mice and was less than the original strain, while also noting the lack of conclusive evidence for a lab leak origin of COVID-19 itself. It really underscores the need to look beyond headlines.